Hello students, today we'll be discussing on a new topic which is the structural organization of a protein. How can you expect the question to appear like? They might ask you to write or describe the structural organization of a protein, which can be an important short essay question in the exam. Okay, we'll see how we have to go about it. Coming to a protein, the proteins are polymers of the amino acids. Which type of amino acids is the L-alpha amino acids, which are majorly present in the human body. The structure of protein is rather a complex and it is divided under different levels. That are 4 different levels of organization. Okay, the protein, once it is synthesized like a linear chain, it will not be present as the same form. It will undergo some modifications, like for example it might undergo some spiral nature, or it might undergo some beta sheet like structures, and finally it will form a complex protein molecule. Okay, it forms a complex protein molecule. So these are at different levels. They will undergo different stage of evolution. Okay, to achieve the final thing, that is the function. To achieve the function, they have to attain some shape. So that shape is achieved by the organization at different levels. So we'll see one by one what are they. Now there are 4 different levels of organization: the primary level, secondary level, tertiary and quaternary level of organization. Okay, a protein will start from the primary level and it achieves till the final. It can be a secondary level, final stage for some proteins. It can be tertiary level for some proteins, and some proteins will achieve the final level, that is the quaternary level. Not all proteins will achieve the final level. Okay, not all protein will achieve the quaternary. Only few will achieve the quaternary depending on its final destination. What is the final function it can achieve with. So all proteins will achieve their final level, which may be secondary, tertiary or quaternary. But total there are 4 levels of organization of a protein. So we'll see one by one. What are they? Okay, primary structure. This itself can be a short answer question in the exam. Okay, primary structure of a protein. The primary structure of a protein is telling us what is the sequence. Okay, sequence of amino acid in a polypeptide chain. A huge chain. Okay, it starts from amino end and it ends with the carboxyl end. This is the starting point. This is the ending point of a protein. Always remember. Okay, so this primary structure will tell us what is the sequence. Like for example, first amino acid is methionine, next is alanine, next is glycine, next is leucine, isoleucine, okay, cysteine. So like this, it will tell us what? It will tell us the what is the sequence. But how they are binding? Okay, what is the bond which is binding them? That is the peptide bond majorly. Okay, the primary structure of protein is maintained by a peptide bond. But if there are presence of sulfur containing amino acids, like for example cysteine or methionine, so they might form sulfur-sulfur bond. So disulfide bond also can be present in the second primary structure of a protein. Okay, a sulfur-sulfur bond, that is a disulfide bond. If you see here also, one should have a clear concept of sequence. Like for example, GAV, that is the glycine, alanine, valine, is of one protein. If you reverse it, glycine, valine, alanine, it becomes another protein. So the sequence will change the type of the protein itself. So sequence is very important, which is defined at the primary level of a protein itself. Who are the bonds stabilizing the primary structure? That is the peptide bond and disulfide bond, which can be important short answer question in the exam. Okay, they'll ask you examples of the primary structure, which includes insulin, gastrin, glucagon, oxytocin, somatostatin, and what are the bonds stabilizing the primary structure? Peptide bond and disulfide bond. Next we come to the secondary level. Secondary level of organization or secondary structural organization can itself be a short essay question or it can be a short answer question in the exam. So here what is happening? there will be some foldings in the linear chain. In primary structure, we have seen it's a linear chain. Okay, it's a linear chain, starting point to ending point. But here there will be some short foldings. Like for example, the protein will fold on itself. This is the first amino acid. This is the fourth amino acid, for example. Now you can see if this was the sequence first and this was the fourth, then they were far from each other. When there is a folding, now what happened? These both have come together or nearby. So what is this? Is it's happening? The foldings of short residues or short chain amino acids segments of polypeptide chain into geometrically ordered unit. The secondary structure denotes configurational relation between amino acids which are 3 to 4 amino acid far. Like for example, first is far from fourth, okay, in linear chain. But when you fold them, when the protein gets folded, they come together, they come nearby. And secondary structure is stabilized by non-covalent bonds. Okay, non-covalent bonds majorly include the hydrogen bonds. Okay, hydrogen bonds will give the stability to the secondary structure. Now, secondary structure arrangements has two further types. It can be organized as alpha helix or it can be organized as beta sheet. So we'll see what is these alpha helix, what is alpha helix and what is beta sheets. Okay, first we'll see about alpha helix, which can be again a short answer question in the exam. So if you see here in the figure also, you can see a spiral nature of the amino acid chain. Okay, a spiral nature. It has spiraled on itself and it will have an axis, perpendicular axis. Okay, it has an axis. So this is the most common and stable orientation. It is right-handed spiral nature and the peptide bond, okay, the peptide bond forms the backbone of this chain. And the R chains, the R groups are extended outwards because if they are inwards, there is no space here. Okay, there is no space within the structure, so it has to be outwards. The R groups are present outwards or thrown outwards. Okay, the structural stability is by the hydrogen bonds. You can see here there are some formation of hydrogen bonds. So hydrogen bonds will give the stability to the structure. It will give the stability to the secondary level of protein, which is the alpha helical structure. The hydrogen bonds are parallel. You can see this is the axis and the bonds are also parallel to the axis of the helix or the alpha helix. When there is each turn, within this, how many amino acids are there? There are 3.6 amino acids making one turn in the alpha helix. So these are the characteristics you should be able to write in the exam. Now coming to beta sheets, the sheet itself is telling it is present as a sheet manner. Okay, like a sheets. So the plated pattern in which R group of the adjacent residues are opposite to each other. If you see in this figure which is drawn here, this is one chain. This is the same chain but on different orientation. The backbone is again the peptide bond. We can appreciate the hydrogen bond stabilizing the structure and you can see the R groups. If the R group is inwards, okay, the pattern of the group of adjacent pattern opposite side, once it is inside and once it is outside, so it is arranged or extended inwards or outwards, the R groups. Okay, and they are composed of two or more polypeptide chains. They might contain single chain or multiple chains. The stability is derived from, again I told you, hydrogen bonds. But here hydrogen bonds are perpendicular. Now for example, this was the axis. The hydrogens are present as perpendicular to the axis. So the hydrogen bonds are perpendicular to the polypeptide chain. Whereas which was parallel in the case of alpha helix. Because in alpha helix the axis is like this and the bonds, hydrogen bonds are formed parallel to the structure. Now beta sheets, based on their orientation, they have two ways. One sheet is called as parallel, other is called as anti-parallel. What do you understand by that? For example, this was a linear chain. Okay, it has folded on itself. So now you can see the direction. This is in one direction and this is in another direction. So what are these? These are orientation of anti-parallel beta sheets. The hydrogen bonds are formed here. Okay, so there is a hydrogen bonds forming to give stability to the orientation, but the movement or the direction of the polypeptide chain is opposite to each other. This is moving to left to right, this is moving right to left. Now if, like for example, you can see, now this is the structure. Okay, now you see hydrogen bonds are formed here. Now both are moving in same directional parallel to each other. Okay, the beta sheets are running parallel to each other. So depending on orientation, they can be called as parallel or anti-parallel. And the bends formed are abrupt U-turns. This you can see, there are U-turns which are stabilized by the formation of disulfide bonds. They are stabilized by formation of the disulfide bonds. Now coming to tertiary level of protein. Now this is the next level to the secondary. It refers to the three-dimensional configuration of polypeptide chain. The secondary structure is further folded, twisted on itself to form a tertiary level. Like for example, you have spiral sheets, beta sheets, again spiral sheets, beta sheets. So like this a protein is formed. A protein is going multiple foldings or twistings to form one protein. So the amino acids that are very distant from each other are brought to very near due to the folding. Like for example, amino acid which is first and amino acid which is 100, because of folding they both are nearby each other. They are nearby each other. Okay, they come nearby each other. It is maintained by what bonds? Apart from hydrogen, they are maintained by ionic bonds, hydrogen bonds, disulfide and van der Waals force of attraction. It is a compact molecule with hydrophobic side chains at the interior. Okay, if you see the orientation of the proteins, the one which are phobic in nature, the amino acids which are hydrophobic, they form the core, and the one which are loving to the water, they are called as hydrophilic amino acids. They form the surface. They form the surface group. What are the examples for the tertiary level of protein? One of the example is myoglobin. Okay, myoglobin is a example for the same. And to get the tertiary and quaternary level, there are set of helpers, or they are assisted by a protein called as chaperones. Okay, these levels are achieved by a chaperone. The final level, the fourth or the quaternary level, is the arrangement of polypeptide subunits in three-dimensional. Now, till now we have seen only one polypeptide chain. Now what happens is there is another polypeptide chain, there is another polypeptide chain. Okay, they all come together. They all come together to form single protein or single multiple protein combination. It exists as protein consisting two or more identical or different polypeptide chain. They can be same one or they can be different. What are the bonds stabilizing? The hydrogen bonds, ionic and hydrophobic interactions. Each polypeptide chain, I told you, if there are 4 polypeptide chains, each polypeptide chain is called as subunit or monomeric unit. Okay, each subunit can be called as subunit or monomeric unit. What are the examples? Like for example, 2 alpha and 2 beta chains of the hemoglobin. If you see the hemoglobin structure also in the any book, you can see 4 structures. That is 2 alpha and 2 beta chains. They all come together to form one chain, one protein, that is the hemoglobin, which helps in carrying the oxygen. If you remove any one of this, it will not be functioning. The protein will lose its function. So examples include hemoglobin, 2 alpha, 2 beta. Similarly, immunoglobulins, which are fighting against the infection, 2 heavy chains and 2 light chains. Creatine kinase, it is made up of only 2 polypeptide chains, dimers. Lactate dehydrogenase complex, it is also containing the 4 polypeptide chains, tetramers. Aspartate transcarbamylase example is example for 6 subunits. 6 subunits comes together to form one enzyme, that is the aspartate transcarbamylase. So different polymer peptide chain coming together to form one protein and to deliver the function. Okay, the function is delivered only in the presence of like this structure. Whole structure should be intact. That is the quaternary level. So to summarize, what are the different levels of protein organization, or the organization of a protein is primary structure level, secondary structure, tertiary and quaternary. To again tell you, not all proteins will achieve the final. It depends on their destination. Some proteins are active at the primary, some at the secondary, some at the tertiary, and some need to achieve the quaternary level of a protein. So with this we'll conclude our topic, that is describing the structural organization of a protein, which is an important short essay question. Some parts can be a short answer question in the same subtopic. Thank you very much.